All right. Tell me when. There, when you're ready. I think, did he turn it on? It's green. That looks promising. It's probably on, right?
Yeah, I think
Okay.
so.
20 seconds. Okay. Oh, should I take my mouth off?
Oh,
Are you, you're going to speak without yours?
Yes.
All right. That's a fancy case, Alex. Okay, we're ready. All right. Good afternoon and welcome to the Department of Medicine Grand Rounds. My name is Evie Slottick and I am an Associate Program Director for the UCSD Internal Medicine Residency Program. It is my great pleasure today to introduce our speaker, who is one of our chief medical residents, Dr. Alex Tong. Dr. Tong attended the Case Western Reserve University School of Medicine, where he was a member of the Medical Scientist Training Program. He earned an MD and a PhD in pathology immunology, where he authored a PhD thesis entitled Lymphocyte Homing, Trafficking, and Function, Insights into the in vivo behavior of regulatory T and natural killer cells. His research led to multiple peer-reviewed publications, including co-first authored papers in immunology and cell biology, oncoimmunology, and frontiers in immunology. He co-authored multiple other intramural and extramural publications. This includes a paper exploring Notch in T-cell mediated graft versus host disease in the Journal of Clinical Investigation, another examining human mesenchymal stromal cell enhancement of hematopoietic stem cell engraftment in hematology and medical oncology, and an investigation into colorectal carcinoma chemokine tumor vaccine in Frontiers in Immunology. During his MD PhD training, Dr. Tong was the recipient of numerous awards, including twice receiving the AAI Trainee Abstract Award and oral presentation at the AAI annual meeting, as well as the Best Oral Presentation Award at the 10th Annual Immunology Training Program Retreat. We were delighted to recruit Dr. Tong to our residency training program, where he participated in the Resident as Clinician Educator pathway and revamped part of the UCSD School of Medicine's immunology curriculum, lecturing on T-cell immunity. Upon graduation from residency, he was selected by his peers to receive the Department of Medicine's annual Humanism in Medicine Award. Dr. Tong was selected to serve as chief medical resident because of his outstanding clinical acumen, his impeccable work ethic, his superlative teaching skills, and his professionalism. And I can tell you firsthand what a delight it has been to work with and learn from this extraordinary individual over the last 4 years. In the interest of rounding out his picture a little bit more, I'm compelled to let you know that he is also not only the progenitor of a fabulous body of research and clinical care, but he is also the father of 2 of the most wonderful children I have ever met. His 5-year-old daughter, Shu Yen, and his nearly 1-year-old son, Kai Yen. He will be speaking to you today on matrix proteoglycans in multiple myeloma, building a biobank to elucidate tumor-immune crosstalk. Dr. Tong?
Thank you, Evie, for that very kind, effusive introduction. It is my great privilege and pleasure to be speaking to everyone today here at Medicine Grand Rounds. And it's been quite a journey, which I'll go into getting here to San Diego. And I'll be talking a little bit about the research I've been doing in Dr. Osmakopoulos' lab. And we actually have some of the lab members here in the audience in attendance today. As far as disclosures, I have none yet. A little about me. So I was asked to share a little about my journey and thought I would highlight the geographic journey first that I took to San Diego, starting from Texas. So as the son of immigrants from Southeast Asia who raised me to love classical music and would take me and my brothers to concerts whenever they had time, the journey here has been marked by symphony halls. So from the Meyerson Symphony Hall in Dallas to the Cannon Center in Memphis, Tennessee, to Severance Hall in Cleveland, where I had my white coat ceremony and med school graduation, to the incredibly beautiful Rady Shell here in San Diego. It's been a musical blast. But the trek through different concert halls has really been in lockstep with my journey in science and longstanding interest in cancer. So starting at St. Jude, when I was an undergrad in Memphis, working in a drug discovery high throughput screening lab, this is where I actually first saw physician scientists taking tumors from kids with cancer in the clinic and walking down the hallways to their labs to try to figure out new treatments, new cures for them. So this inspired me to pursue MD PhD training at Case Western. So I ventured to Cleveland, and this is where I fell in love with immunology and where I met my wife, Sherry, in medical school, who single-handedly stopped my nomadic existence as a naive T cell and has helped me mature progressively over the years. Then upon finishing training in Cleveland, Sherry told me, go west, young man, but specifically San Diego, because that's where I'm doing neonatology fellowship. And I was incredibly blessed to have matched here at UCSD for residency to join the family that Dr. Jassel and the APDs have nurtured now through a pandemic and find an amazing research mentor and a physician scientist role model in Dr. Asimakopoulos. But what would a journey be without a bump in the road? So in the middle of residency, actually in the middle of my research block, I was diagnosed with a germ cell tumor, and I found myself on the other side of the exam room. And I had the proverbial notebook in hand, hanging on to every word that my UCSD cancer team was saying to me. And after a visit to the mechanic, I am super happy to report that I'm all patched up, back on the road. But what reassured me in those darkest hours, aside from all that incredible love and support my family, the residency, the lab gave me, was that my cancer was quote unquote curable. But unfortunately, we don't have a cure for many other cancers, including myeloma. And so we've got a lot of work to do. And that's why I want to be a medical oncologist when I finally terminally differentiate. So here's an overview in sonata form of what we're going to cover today. A brief intro on myeloma, some exposition then on our lab's overarching research on matrix proteoglycans, a little bit of development on building a biobank, and then a return to the main theme with some thoughts on where we go from here. So some PMH on myeloma. The first well-documented case came out in 1844, and this described a rather unfortunate 39-year-old woman who developed severe back pain. She was prescribed rhubarb and orange peel, but then she died 4 years later after onset of symptoms. And her postmortem exam showed multiple fractures, radius, ulna, femurs, tibula, fibula, and the replacement of her bone marrow with a red substance, which we now know to be multiple myeloma. And our understanding has progressed rapidly over the years. We now understand this disease as one of terminally differentiated plasma cells, which primarily reside in the bone marrow, but can also be seen in the peripheral blood as well as other tissues. And the monoclonal protein produced by these plasma cells in combination with the malignant cells themselves and elaboration of cytokines really drive end organ damage, which we now know as the CRAB features, hypercalcemia, renal insufficiency, anemia, and bone or lytic lesions. But a really nice analogy for how to think about myeloma is one I picked up from Dr. Asimakopoulos. So here, plasma cells are trained snipers, if you will, and they're shooting out antibodies. And you can think of these as the bullets of the immune system. Normally, they're pretty specific, and they're used to kill invaders while leaving our healthy cells alone. But in myeloma, when plasma cells go haywire, there are just way too many snipers, and there are way too many antibodies. And so you have indiscriminately progressive collateral damage to your healthy cells, and this really leads to the cancer's protean manifestations. All right, so I just wanted to highlight some of the history and major milestones in myeloma from that first case report in the 1840s to advances in diagnosis and staging with more molecular tools that we're familiar with today. And all of this has been happening concurrently with advances in treatment with chemotherapy, autologous stem cell transplant, and small molecule inhibitors. But we are now living in the era of immunotherapies, We have monoclonal antibodies that are targeting myeloma antigens. We have bispecific engagers, chimeric antigen receptor T and NK cells. And as you can see by all of these recent articles in a highly reputable journal in the past year, it's really an amazing and exciting time to be in medicine. So a little bit about myeloma by the numbers. Well, it's the second most common heme malignancy after non-Hodgkin's lymphoma. There's about 34,000 new cases per year and 12,000 or so disease-specific deaths. The 5-year relative survival, it's about 58%. And this has been improving over the years with many of the therapies that I've mentioned. But the major problem is that a true clinical cure remains elusive. So we can see here the progress that's been made. But what I remember being so frustrated about in clinic, seeing a new diagnosis of multiple myeloma and being part of that patient encounter is telling them that this is not a curable disease. You can think of it as something that'll temporarily get beaten down, that you can temporarily chase away, but kind of like your first breakup, it'll eventually come back to haunt you. So let's put this into the context then of a popular way to think about the evolution of the tumor host immune response, and that is cancer immunoediting, as envisioned by Bob Schreiber at WashU. So in this framework, we have 3 stages that are described following the initiation of cancer. The first is elimination, where you have a competent immune system that is able to successfully kill tumor cells. But then you can run into equilibrium, where there's tumor dormancy in the face of selective immune pressure. And then we get to escape, where you have a successfully immunoedited tumor that escapes the immune system and results in progression. But intimately tied into all of this is the extracellular matrix. And this is a diverse non-cellular 3D network of proteoglycans, glycosaminoglycans, and fibrous proteins. And the matrix is dynamic, and it's an integral part of the tumor microenvironment. And yet, its contribution to this cancer immunity cycle is just beginning to be explored. What I want to highlight here is that there's a complex interplay between this extracellular matrix and the components that we are very interested in and trying hard to understand. So the part of the extracellular matrix that our lab focuses on is Versacan. And this is a large matrix proteoglycan. It's got crucial non-redundant roles in organ development and disease. Knockout of this is embryonic lethal. It has a lot of roles in cardiac development in particular and kind of interdigital formation. So there are a lot of sources of Versacan in the tumor microenvironment. This could include tumor cells and stromal cells, but especially tumor-associated myeloid cells. And increased Versacan expression has been documented in several malignant tumors to be associated with relapse and poor prognosis. Now, Versacan undergoes regulated proteolytic cleavage into a bioactive fragment called Versakine. And this itself has immunostimulatory properties. So we're going to talk a little bit more on this seemingly opposing dynamic in a minute. I'm going to continue with introducing a few other players in the tumor microenvironment. So number 1 would be dendritic cells. So back to immunology 101 in medical school. But these are the orchestrators or professional antigen-presenting cells of the immune system. And they're kind of like the drill sergeant that tells the poor naive T cell to grow up and to start fighting. Then we have our macrophages. So these are the big eaters, kind of like the Pac-Mans of the immune system. They also have multifaceted roles, though, in immune regulation and in wound healing. And several studies have now shown that this large molecule, intact Versacan, has immunosuppressive effects in the microenvironment. This can result in what we term tolerogenic dendritic cells or suppressive macrophages. But on the flip side, when we start to look at the action of Versakine, this cleaved fragment of the parent molecule, this has immunostimulatory properties in several cancers. So for example, Versakine can promote the recruitment, activation of conventional dendritic cells. And these then stimulate some of our CD8 positive T cells, cytotoxic T cells. And this occurs in different tumor types and backgrounds. So here we have a fascinating, seemingly opposing effect of the parent molecule, Versacan, and its cleavage product, Versakine, in the tumor microenvironment. All right, but as we wrestle with why some cancers remain incurable, we also start to think about this idea of immune exclusion in the tumor microenvironment. So you could have T cells that are trying their best to gain access to a tumor, but for whatever reason, whether it be stromal barriers or some of these other listed factors at play, these T cells are exhausted, they're excluded, or like when you forget your wedding anniversary, they are excommunicated from the tumor bed. Now, there's also evidence that this exclusion happens to dendritic cells. And so these, again, are the important drill sergeants that are telling the T cells to wake up and to find their targets, but are instead being separated from these soldiers that they're supposed to train. So our lab is trying to ask the question, what is the stroma doing to these important immune cells? And in particular, how might elements of the matrix like Versacan regulate dendritic cell and T cell behavior in tumors? And we started out with the observation that the parent molecule, this intact Versacan, actually accumulates and undergoes cleavage in multiple myeloma marrow. And you can see here increased staining compared to normal marrow, where you don't see as robust staining. Around the same time, we also had evidence by others in the field showing that Versacan, this big molecule, signals through a toll-like receptor, and this can promote dendritic cell dysfunction, kind of turning off those drill sergeants, if you will. And it causes these cells to produce immunosuppressive cytokines like interleukin-10. And you can kind of think of this action as not a good thing in the tumor microenvironment. Follow-up work by our lab showed that Versacan proteolysis, so formation of this fragment, Versakine, at the time of marrow recovery, and we were looking at initial autologous stem cell transplant, this actually correlated with increased CD8 T cell infiltration into the myeloma bone marrow. So as you can see here, from left to right, the more Versacan proteolysis, or Versakine, that fragment that we detect, the more CD8 T cells that we have in the marrow. So this cleavage product, we also know, induces inflammatory signals in vitro. And so one way that you can kind of think about it is the more Versacan proteolysis you have, the more CD8 T cells you have in the tumor, probably a good thing at this stage. And interestingly, this association with cytotoxic T cell infiltration is not just restricted to myeloma, but we also see this in solid tumors, such as colon cancer. So the more Versakine that is present, the more CD8 T cells are in the tumor. And just to note, in colon cancer, this is actually regardless of mismatch repair status. We've also looked in lung cancer as well, and we see a similar association of this cleaved product and CD8 T cell infiltration. And we also see an association with favorable prognosis. With the more CD8 T cells you have, the better that these patients do. So I just wanted to stress that this is a phenomenon that's not just restricted to myeloma, but has prognostic implications in other tumor types. But going back to myeloma, when our lab tapped into the biobank that was available at the University of Wisconsin-Madison, where Dr. Asimakopoulos hailed from, we found that this cleaved product in the bone marrow after autologous stem cell transplant actually correlates with worse overall survival in multiple myeloma. So this represents kind of a paradox. So we have intense Versacan proteolysis into its supposedly immunostimulatory fragment with concomitant CD8 T cell infiltration, actually leading to worse and pretty dismal post-autologous stem cell transplant survival. And so from this, we can surmise that there must be some immunoregulatory or suppressive networks that are rendering perhaps these CD8s or other elements of the immune system dysfunctional. And so our challenge then is to figure out why. So what I'm going to do is just briefly touch on some of the various tools that we have to study the immune response in multiple myeloma. To start out with, we have many cell lines that are available. And these are kind of contrived in vitro systems where one can add a stimulus or co-culture with immune cells, for example, and you get an idea in vitro of what's happening. And we also have mouse models on top of that, which our lab has actually been a pioneer in myeloma. We've made one of the more faithful myeloma mouse models to date, kind of in a RAS-driven manner. But at the end of the day, despite having all these cell lines and better mouse models available, there's just no match for primary patient samples, obtaining them and learning from them. And we've had some lessons we can draw from the Cancer Genome Atlas, although notably left out myeloma. But with this in mind, our lab sought to build and leverage a disease-specific biobank. And so let me go a little bit into what this entails and why this is such a priority for researchers. And just of note, this was a priority too for the Cancer Moonshot Initiative. Okay, so I've learned a lot over the past couple of years. And the major takeaway here is that building a biobank ain't so straightforward. You need buy-in from multiple parties, including clinical coordinators, nurses, faculty, pathology colleagues, lab personnel, and most importantly, patients. And so just kind of to start out with the workflow of how this might occur, we start with an IRB. And so this can take a couple of months for review, revision. But once it's approved, we then need to engage clinical coordinators and physicians about approaching patients to participate in sample collection. And when an appropriately selected patient is identified, for us, it's going to be the initial autologous stem cell transplant setting. They need to be seen in clinic, and informed and consent then needs to be obtained to participate in the study. Then at the appropriate clinical time point, samples will be collected, in our case, bone marrow, plasma, and laboratory personnel need to be notified to actually process the samples. And then after processing and storage and initial analysis of these samples, cataloging is done. And then from here, samples and clinical annotations can be extracted for analysis. So from this workflow, I hope you can appreciate the myriad parties involved. And of course, there's going to be a couple of challenges along the way. So let's talk about a few of them. I think they're worth mentioning. But first, a shout out to our amazing lab manager, Alejandro, who is here in the audience, who has basically carried, like Atlas on his shoulders, the efforts forward to try to build this biobank. So number one, if your project happens to be on a parent IRB, and there is a change to that IRB, and you have all these amazing consent forms that are printed out in multiple languages for your patients, you then have to revamp all of those forms, and this can lead to a delay in collection and consent. Identifying eligible patients for the study can also be challenging. So how do you coordinate when they'll come to clinic and when you'll have a team member who's actually ready to help with the consent? And if coordinators who are helping you are actually working on multiple projects, that is, you don't have a disease-specific coordinator, their bandwidth may be kind of limited. And then on the day of collection, laboratory personnel need to be on call to process the samples immediately, and pathology needs to receive samples for processing. And just to give you an idea, the processing time can take anywhere from 3 to 4 or more hours for each round. Now, we have protocols in place for determining sample yield and viability, but you can see variability in how much or how little samples obtained patient to patient. We're super fortunate to have an amazing working relationship with our pathology department, in particular Dr. Huanyo Wang, but requesting assistance with tissue processing and prep also takes some time. Data safety and security is another important consideration, and having access to encrypted repositories for data storage long-term is important. And then finally, when these samples are stored for the next couple of years at -80 or liquid nitrogen, who's going to pay for the upkeep and who manages the inventory? So in summary, a lot of moving parts and potential pitfalls, but it's also a great way to build relationships and collaborations from clinicians and basic scientists all across the institution. All right, so a little bit about how our biobank is doing. Here's how it's been growing since it was started. We're now approaching about 24 patients for whom we have bone marrow core biopsies and aspirations post initial auto stem cell transplant. We have viably frozen bone marrow as well as plasma. And these are some of the staining that we can do to recapitulate some of what we've seen before. Kind of the staining patterns, low to moderate to high in the tumor microenvironment, and really harkening back to our biobank that we had available to us at Wisconsin. But we can talk about leveraging a biobank in other ways and trying to look at clinical outcomes. And although the data are immature, given the longer length of follow-up that's needed, we're already starting to see differences in survival probability when using Versican proteolysis moderate or high as a biomarker. And so this is in line with our prior findings from the biobank that we had in Wisconsin. But now that we have access to higher quality samples and viably frozen tissue, we can start to interrogate immune pathways with newer technology, such as nanostring, which I'll briefly introduce here as a multiplex nucleic acid hybridization technology. This uses unique optical barcodes to enable digital counting of individual RNA molecules. So you can take samples from our biobank that have different Versican proteolysis patterns, and you can subject it to this analysis. And by doing so, we can start to explore some of the immune regulatory genes that we're hypothesizing are significantly different, and then use this as a launching pad to dissect this tumor-immune crosstalk. All right, so these are the kinds of data we're now generating, where each well represents a different gene that's up or down in expression in a patient with high Versican proteolysis versus one who has low proteolysis. We're now validating and exploring many of these genes, so I won't share specifics here, but generally, what I can say is that many of our hits are intimately involved in immune regulation. They can offer mechanistic insight into matrix-kind function, and importantly, some are even targets for intervention. And the hope then is to uncover new pathways in cancer, as we've done exploring the Versican conventional dendritic cell axis in solid tumors, leveraging the TCGA database to help us see where this pathway might be relevant. And finally, the power of having viably frozen bone marrow and plasma is that we can run things now like multiparametric flow cytometry, sorting individual immune and tumor cells for further analysis. We can even start to characterize cytokine levels and perform functional assays. And this really offers us the ability to validate some of those targets from the gene panels and better tease apart the biology. So I hope from all of this, you can appreciate now how having primary samples is critically important to us as we try to figure out what's really happening in the tumor-immune stromal axis. All right, so to bring us back to where we are now, as we start to realize the amazing possibilities of novel immunotherapies, these therapies have to work in a relatively immunosuppressive environment and unfavorable conditions where we know that matrix proteoglycans like Versican, Versikine play important roles in shaping the immune response. And then by using a biobank on top of our existing in vitro approaches and mouse models, we can start to unravel how to leverage this extracellular matrix to our advantage. And the hope then is to possibly interrupt this cancer immunity cycle. This has implications, again, not only in myeloma, but in solid tumors, where we know that Versican and Versikine have distinct activity. And so there's tremendous potential to be unlocked in terms of breaking down barriers to tumor elimination. I'll share that in terms of the next steps, we're very interested in the modulation of dendritic cell behavior by the matrix proteoglycans, having recently explored this in lung cancer. And we'd also like to explore what's going on with matrix proteoglycans and relapsed refractory disease. Still remains understudied. But ultimately, to bring us back to why we're here, the hope is to move the needle a little bit closer to a true cure for myeloma, and a biobank is a step in that direction. All right, so I'd like to close with some inspiration from a thought leader and the prior head of the NIAID Laboratory of Immunology, Bill Paul, the late Bill Paul. He was a mentor to many of my scientific mentors, and I remember running into him at different meetings in immunology, and he always took time to talk with young scientists, although he himself was a giant in the field. But he eloquently described in this article a career in science, and particularly immunology, as one of endless fascination. And as we appreciate more and more the immune system as a wonderfully complex symphonic dance, if you will, with myriad variations and counterpoint in health and disease, I couldn't agree more. And I hope that you can see why. Okay, so I'd like to take a little bit of time as well to thank a whole bunch of people But first, Dr. Asmakopoulos, who was recently inducted into ASCI, and members of our lab, Alejandro, for again, carrying this biobank work forward. Thanos, our amazing physician scientists, Yoon and Duncan, and more members of the lab present and past. A heartfelt appreciation to our wonderful BMT coordinators and faculty for their help and collaboration, other collaborators in pathology, extramural colleagues helping us work on other biobanks, and then my incredible UCSD IM family, including Dr. Borak for her support of resident research. We have various funding sources, and I'd be remiss if I didn't thank my amazing wife and my kids who find some way every day to tickle my marrow. And for our attending lifelong learners, don't forget to get your CME here, the barcode at the bottom. And with that, I'll just say thank you so much for your attention. Happy to take questions.
Dr. Tong, thank you for a phenomenal talk. It was edifying, it was clinically relevant, it was accessible even to a non-physician scientist, and it was filled with cutting metaphors. I don't think there's more that we could ask for in a Grand Rounds talk. So Dr. Tong's presentation is now open to questions. I don't know if you all want to start.
I'm sorry if I missed it, but do you have any, is there any insight as to why the folks who had more peptidoglycan or the varic, I'm going to say all these wrong, the varicase into varicine as why they had worse outcomes? I feel like
Yeah. So no, it's a great question. The question is, what's the mechanism by which perhaps these fragments in the tumor microenvironment is leading to worse outcomes? I think the short answer is we don't maybe have like a smoking gun right now, but some of the preliminary data that we have from the gene panel analysis does show that there are a number of more immunosuppressive genes, molecules that we kind of have already classified in literature as dampening down the immune response. And so what we're doing in validation assays, kind of going back from this bedside to bench model, is trying to validate some of those pathways. But you're absolutely right. For us, the really interesting thing is that it's a paradox when we look at other models, like in lung cancer in particular, but even like in vitro, this bioactive fragment is supposed to be immunostimulatory. And yet, when we see more of it in the tumor microenvironment, in myeloma in particular, it's not leading to better clinical outcomes. I will say that there's this also maybe kind of helpful view to take, which is the relationship between Versacan, this parent molecule in development, and kind of forming like a loose sort of matrix that is very permissive to cells moving in and out. And then as development occurs, it compacts and it forms, something that's maybe a little bit more, we think of kind of like a barrier. And so in the tumor microenvironment, what you have is like cleavage, of this big molecule that, is occurring. And yet we have all these fragments that are also at play. So I don't know that we have a clear answer yet as to, which of this kind of like yin yang balance, is kind of winning out. But this to us was such an interesting clinical conundrum and hasn't really been looked at so far. But yes, you can hopefully, when we put all the pieces together, you can look forward to it in a publication. Okay, great. Yeah, so it looks like we had a question from Margaret. So I mentioned that finding CD8s in colon cancer specimens is associated with favorable prognosis. Might it be a common practice to look for CD8 T cells in this setting, and what might be implications for treatment? Yeah, I will say that in many different tumor types, and maybe more so in solid tumors, the presence of infiltrating T lymphocytes generally is kind of a good thing when we can use this prognostically to maybe predict who might respond to treatments. And some of the scenarios that I kind of look at are kind of immune checkpoint blockade. And we have scoring systems, for like PD-1, PD-1 expression. But as we kind of think about tumors that are a little more immunogenic, we would expect to see maybe more CD8 T cells and maybe more of a response to some immune therapies. And so it's just kind of curious again here that we see a paradox in myeloma. It's not quite like maybe some of the other solid tumors that we're used to seeing and treating, and hence, worth more investigation.
All right. So thank you again, Dr. Tong. For those of you that don't know, we are fortunate enough to get to keep Dr. Tong for another period of years and hopefully forever. Yay! He's going to stay on to do, not surprisingly, HEMONC fellowship at UCSD. So look for him in a lab, in your favorite journal, and on the wards in the years to come. Thank you so much, Dr. Tong.
Thank you.
Amazing.
Thank